Scientists or not, we are all familiar with the concept of genetic engineering. Whether it's our moms telling us to avoid cookies with genetically modified ingredients, or the scientists using frog DNA to bring back the dinosaurs in Jurassic Park. But it turns out the biggest discovery came not from a T-Rex, not a cookie, but a germ. CRISPR is the cheapest and most effective form of genetic editing technology. While it is still too expensive and complex to do on your kitchen table, it's cheaper and faster than the gene editing technology that came before it. So what do we know about CRISPR, other than it makes me really hungry for a Krispy Kreme donut? Well, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Basically, scientists discovered CRISPR DNA in bacteria. Now, bacteria don't have an immune system like humans. They use CRISPR DNA as their own form of immune system. When a virus infects bacteria, it inserts its own DNA into the bacteria's DNA. Here's where CRISPR becomes useful. The bacteria has a special mechanism, a nuclease protein called Cas9, whose purpose is to create a double-strand break in DNA. The protein needs a special form of RNA called guide RNA, which guides the Cas9 protein to the location of the bacteria's DNA where the foreign DNA is located. Think of this pair of scissors as the Cas9 protein, and my hand as the guide RNA, showing the Cas9 scissors where to cut on the delicious DNA. The DNA will begin to repair itself, usually by just shoving the two segments of DNA back together. This renders the foreign virus unable to cause harm, but its genetic material stays in the genome as a reference for when the same virus attacks, similar to how our bodies become immune to a virus after getting sick with it once, thanks to antibodies. The part of this process that caught scientists' attention actually wasn't the CRISPR DNA, but instead the Cas9 protein and guide RNA combo. This is because guide RNA is significantly easy to program, which means CRISPR technology could be removed from bacteria and programmed for any genome at any section that you want. This opens up endless possibilities. Scientists are learning which genes express what traits. They do this by cutting specific sections of a genome and figuring out what function stops. Some scientists are visualizing the genome by attaching a fluorescent protein to Cas9 so that they can highlight specific locations. Scientists can also insert segments of DNA between the genome by tricking the DNA into repairing itself onto the end of the inserted strands. CRISPR technology has opened many doors. Genetic conditions that previously we couldn't touch now have potential for a cure. Researchers also recently found a way to turn off the immortality switch that causes cancer cells to reproduce indefinitely in brain cancer. CRISPR also has applications in the food we eat. Scientists are able to edit genes to make plants like tomatoes grow much larger than before. CRISPR still has hurdles to get over before it can have an impact on things like medicine. Questions like, how are we going to edit the genome of a complex organism when CRISPR technology only works one cell at a time? Not to mention what the ethical limits of CRISPR should be. Whether it would be right to use CRISPR technology for things like embryo editing or cosmetics. But no matter how we use CRISPR technology, it is bound to change the world of biosciences. And it sure tastes delicious.